my name is Tracy Hong, and I will be talking about the interest of the 3D material that mimics nature to transform cells to muscles. So, a little background information is that this was developed by Dr. Richard Williams along with other biomedical engineers at the Royal Melbourne Institute of Technology. These researchers have successfully incorporated the natural processes of embryonic development to build a material that can naturally communicate with stem cells for effective tissue repairs. The 3D hydrogel scaffold was created by a combination of lab-made peptides with natural proteins and along with other polymers. Overall, this process is to provide hope for people who suffer from injuries where their muscles cannot repair themselves anymore due to trauma or disease. Also, this helps the rapidly aging population as it is a new developed way to replace and repair the parts of the bodies where they are worn out and damaged over time. Finally, this process is not only cheap and simple, but is very powerful. And now I will move on to Jacob, and he will be discussing about the current state of knowledge.
My name is Jacob Guerra, and I have the current knowledge in the hydrogel scaffold. So hydrogels are hydrophilic polymers, and when trying to make tissues, they should have the same properties as the body cells so they can carry out their functions to reproduce. So these gels share many similarities with the body cells, allowing them to cooperate with one another and communicate with the stem cells to replace the damaged tissues. So now we have a lot of technology to be able to integrate different types of cells within these gels. And there's not only one gel, but there's different chemistries and different types of control of different cells and gels to form tissues. So light and temperature can modify the gels to encapsulate the cells and force them to communicate with one another. So like for example, using liver cells and fibroglass cells, they can be controlled to allow communication to help each other become functional within the body. They assemble the cells to mimic tissue structures. For example, you can use heart cells and modify the gel to mimic the shape of the desired heart muscle tissue that you want to, and replace the affected area. So like this concept would apply to like all the different types of tissues they want to create, but the procedures do vary depending on the size of the tissue they want to replace or the cell types and just what functions they carry out.
Hi, my name is Elizabeth Salamanca, and I'm here to talk to you about material that can mimic itself to transform cells into muscles. Did you know your muscles can regenerate when necessary to a certain extent, such as small injuries? However, when we get into certain situations, we may not be able to. For years, researchers have been trying to understand how cells work and organize in 2D. Well, in Melbourne, Australia, several biomedical engineers led by Dr. Richard Williams found that lab-made peptides with proteins and polymers can create a 3D hydrogel scaffold. Hydrogel-based scaffolds can change their characteristics to be similar to natural tissues. This material is more likely to interact with stem cells for tissue repair. The stem cell will then be able to turn into muscle fiber, therefore aiding muscle regeneration.
So this is Chase Bowling, here to talk about the next part of our project. And as you already know, for you potential investors, is the fact that it uses a compound to regenerate lost tissue in hopes of rebuilding a human damaging. The main problem is that the compound itself requires the use of stem cells. Now, for those who are not quite sure what those are, it is embryonic tissue used mainly in the building of a body when it was in the embryonic state. Main problem is it has to be human tissue for it to be compatible in our compound, and that means we have to extract it from a living embryo. Main issue here being that there is currently no process that exists that would allow the embryo to survive such a process. Now, under law, this is not an illegal practice, and in fact is perfectly acceptable in many countries. The main issue is, from an ethical standpoint, many people are not approving of it, seeing it as a possible form of murder to the embryo. Now, this is going to be the biggest issue in moving forward with our project, as regardless of how much good we can do with our compound itself, there will always be public outrage over how it is created. That is, of course, until either the future where we might be able to synthesize this compound either from a completely artificial source or possibly even from a combination of animal stem cells, or maybe even possibly finding a way to harvest said cells without causing any damage to an embryo. Until these can be found, we cannot say we will be 100% approved by the public, but the process itself is still legal and would be effective for most people. This would be Chase Bowling, signing off.
Hello, my name is Tristan Villa, and I'm here to discuss where things are going on our topic of muscle development using a lab-made 3D hydrogel scaffold. This discovery is going to possibly, in hopes of, help those that are injured and suffering from injuries that disallows their muscles to repair themselves. These are usually caused by traumas or disease like muscular dystrophy. We can also take into consideration that our population is aging. As our body grows older, it is obvious that it breaks down and becomes less efficient. Therefore, this discovery of using stem cells and the 3D scaffold could help with many muscle issues in the future. Another point on where things are headed is that researchers began to explore the possibilities of making a bioprint of these muscles with stem cells already embedded and then joining in with nerves, blood vessels, and other muscles to then make these spare parts that we could use. So in conclusion, at one point in the future, we may have a time where we could replace old dying muscles that are no longer working due to these traumas or diseases and completely replace them with lab-grown synthetic muscle almost.